If you had one shot, or one opportunity to seize everything you ever wanted in one moment.
Menarguez here, the head coach of Team Mitosis, as we take a inside look at Team Mitosis.
Well, our team is starting off in preseason, or some like to call interphase. This is where we spend about 90% of our time trying to get ready for the season. Last month, we were in the G1 phase. This is where we need to make all the things getting ready for DNA synthesis, getting ready to duplicate the DNA. This is where the cell continues to grow. Right now, we're in the S phase with DNA synthesizing. Now the core of the team is solidified. Next month, we're going to look into the G2 phase and make sure all the other players are ready to go, such as replicating the organelles and getting them ready for cell division. The only thing we need to focus on this year is the GO phase. We had a lot of players out due to injuries, and they were placed in the GO phase because they were unable to divide. And that's pretty much a wrap-up for our preseason.
We just got a new injury report about a liver cell. A liver cell is one of the top players for Team Mitosis, and this liver cell did not pass the G2 checkpoint. This just happened a few seconds ago. We just got word, but now it's not active and is not dividing, and now will be on the injury reserve. Back to head coach Menarguez. Thanks for the update.
So, what are your key objectives for this upcoming season?
The season is short but filled with different stages. We're starting in the prophase, where the nuclear envelope starts to dissolve and the chromatin starts to condense. Then we enter pro-metaphase, where the mitotic spindle starts to form. The middle of the season is where the action starts, and most of the fans like to see, as the chromosomes line up along the metaphase plate. Anaphase is later in the season as the chromosomes separate. At the end of the season, at the very end, our team goes through telophase, and this is where the chromosomes migrate towards the poles of the cell. Then we get into the playoffs, the real crunch time. And this is when we have an animal cell. So we are using cleavage furrow, which cuts the cells into two daughter cells, creating two daughter cells. Thank you for having me on ESPN Insider. Thank you for having me.
New York City. You are now rapping with 50 Cent. You gotta love it.
This is ESPN Insider Look at Team Meiosis. This is head coach Ryan Egan.
We start with our preseason, and we actually stole our preseason blueprints from Team Mitosis. Don't tell head coach Menarguez.
Coach, what are your upcoming objectives for the season?
For Team Meiosis, we've broken the season into two parts. Meiosis 1 is the first part where the homologous chromosomes are separated. Our season is similar to Mitosis besides the fact that in prophase 1, crossing over occurs and a tetrad is formed from the homologous chromosomes. In metaphase, the tetrads line up and then are split as the sister chromatids remain intact. Then telophase occurs similar to Team Mitosis, forming two haploid cells.
This is ESPN Analysis here, and we're trying to break down what head coach Ryan Egan was trying to say. We want to emphasize to our viewers that in prophase 1, a tetrad happens, and this is when crossing over occurs, and this allows for genetic differences between the daughter cells. We also want to emphasize that in meiosis 1, it's the separation of homologous chromosomes instead of the sister chromatids. Back to Ryan Egan. Coach, you talked about a two-part system in your season.
Yes, there's a second part of the season where the sister chromatids separate. Now we look into playoffs where it is crunch time, as cytokinesis creates 4 haploid cells.
Oh, perfect. Thank you for giving us an update on ESPN Insider. Thank you for your time, Ryan Egan, and good luck this season.
Thank you.
Well, that's a wrap for ESPN Insiders. We want to take the time to say thank you to Lafayette College, Professor Drummond, lab TAs, Gina, and Andy. Tune in tomorrow at 8 p.m. Central for a 30 on 30 on the history of mitosis. Good night, everyone.